Hello, Dr. Ellis here, and we're going to go through prokaryotic cell structure and then compare some other bits about cells as well. Um, so if we just zoom into, these are the words that we're going to label on this diagram: cell wall, etc. So let's just have a look at this. This is like a generic bacterial cell, a prokaryotic cell. So we're just going to pop around and label this. I'm going to start on this here because here's our cell membrane. And on the outside of the cell membrane, we have a cell wall. And we'll be going through what that cell wall is. It's not made of cellulose. Plant cell walls are made of cellulose because they have lots of sugar because of photosynthesis. So if you do photosynthesis, you tend to build stuff out of cellulose because it's a good structural molecule. Whereas here, bacteria don't have access to lots of glucose and therefore they build it out of something else. So that's our cell wall. And inside the cell here, this is just cytoplasm. Incidentally, this is the cell membrane. It's the only membrane that it has. Coming around here, let's go to here. This is our flagellum. Sometimes they have more than one. Over here are our ribosomes. All cells have to have ribosomes. If you've got DNA, then you need to have ribosomes. Here's our loop of DNA. And it's not the only DNA it has. It also has these two things just to the top left. These are called plasmids. People mislabel these as lysosomes or vesicles because, quite frankly, if you're drawing them, you draw them in the same way. This kind of structure here, the last one that's labeling, is called a mesosome. Mesosome. I'm going to put that in little quotation marks because that might not exist, and we'll see why later. And then we're going to draw on the last thing, which is the capsule. So around above the cell wall, so the cell wall's down here. So we're just putting on like a wobbly layer. Again, I could have added it, but it's awkward to draw. Obviously, it's not awkward to draw now. It's awkward to draw when I'm trying to make a clear diagram using PowerPoint. So this is our capsule. It encapsulates it. I'm just going to put a little asterisk next to capsule. I'm going to put a little asterisk next to flagellum as well. And yeah, super. And it's just saying that these are not found in all bacteria. So all bacteria have everything else, but they might not have a flagellum and they might not have a capsule. So that's just our labelling. I've put a scale bar on here, but it's about the same size. Typically the same size as a chloroplast or a mitochondrion. You do get a few like gigantic bacteria, and you get some really tiny ones as well. So we're just going to go through a few things about it when it's relevant. And the first one we're going to get to is cell wall. So it's important that we know what the cell wall is made out of. So it's made out of murin. So it's made out of murein, murin, and that's a peptidoglycan. But the word on the syllabus is murin, and it does the same job as any other cell wall. It kind of helps prevent the cell from bursting. It gives it structural support. Cell membrane is the same as every other cell. And it's a future topic, is this? It's just so we call it a phospholipid bilayer. Again, it won't mean a lot at the moment, but we're going to be meeting about phospholipid bilayers. The capsule, again, the interesting thing about it is that it makes you more likely to be a pathogen. It's harder for a white blood cell to engulf you if you have a capsule. So we're just going to put here that bacteria that have a capsule. So hopefully you kind of following along. pathogenic, which is a disease, which is disease causing. So you see pathogen at GCSE. Flagellum, this just allows it to have powered movement. Some bacteria with flagella. So flagella is plural of flagellum. You don't lose marks there. Plural and singular. Um, we call it motile. M-O-T-I-L-E. They have powered movement. So there's some bacteria that will swim in the soil. In like the air spaces where there's water in the air spaces, they'll swim and they'll like move into plant roots. A famous, one with the flagellum is the one that causes cholera. Again, that will like swim into the mucosal lining in your small intestine and burrow in. Again, and some like E. coli, they don't have flagella. Yeah, so some do, some don't. It's just knowing, like a table of differences, etc. Plasmids you met at GCSE. This is a small ring of DNA. It's exchanged between bacteria. So bacteria don't do sexual reproduction. They don't make sex cells, but they do exchange a bit of DNA between them. Often these plasmids are, have copies of like antibiotic resistance genes and things. So, we manipulate plasmids in that we can slot new genes into them, and therefore we can move genes into bacteria. It's not that important, these plasmids. It becomes important in topic 8, end of year 13. Just know that they may have them for now. Next is our 70S ribosomes. So these are a smaller ribosome. Than the 80S found in eukaryotes, karyotic cells. Again, that's a bit of a lie, because remember, mitochondria also have 70S ribosomes, but they are within an organelle within a cell. So so smaller ribosomes, and they still join amino acids. These so I can just put that they carry out protein synthesis. So they do the same job. It's just evolution for you. Bacteria came first, and eukaryotic cells came second in terms of life, and the 80S ribosomes have been added to. It's not just they don't do anything different, but maybe they're a bit more efficient or something. Then when we get to our loop of DNA, this has the genes of the bacterium. So so it kind of forms the main genome. I mean that most of the genes. Oops, that's trying to be an M. Try that again. Yeah, most of the genes. Remember, a gene is a section of DNA. Most of the genes are here, because some of them are also in the plasmids. And it's not wrapped around these things called histones. So just put that down. So it's not associated with histones, like a protein that ours are because we're a eukaryote, we like spool our DNA around histones. And it all comes from the fact that if you're a single cell, you've no need to like turn your genes on and off for like long periods of time because like the cells are not going to specialize. Whereas we're multicellular. I want a muscle cell to only have muscle genes turned on, and therefore they wrap all the other genes that they're not going to need, like for making the retina or something. All those are wrapped away, therefore they are effectively turned off. Cytoplasm is cytoplasm. So it is. We can't really move on from key stage 3. Yeah, it's where chemical reactions occur. Then we get to mesosome. It's now in brackets rather than inverted commas, but never mind. We used to think that this was a place, so it was like the Christie membrane, as in a way that they can get a larger surface area. We now think it's probably an artifact, as in when we've looked at them under a microscope, we've dehydrated the cell slightly, and therefore it's just like a shrunken bit of the cell membrane. So that's what it really is now. So a mesosome, it's most likely. Oops, pick a pen. Yeah, so it's most likely an artifact. It's a leftover structure due to dehydration for viewing with a TM. It's not on the syllabus anyway, so it doesn't say you need to know what mesosome is. But again, the question they could ask you, they could say, like give you the information, say it's a mesosome, and then ask you why it's likely to be an artifact. Remember, bacteria are tiny. You wouldn't even really see them with a light microscope in school. You can see them as like little specks, but you need to have a times 100 objective lens. So you need to begin to times 1,000 magnification before you really start to see bacteria under a light microscope, and then you would just see them as like little specks. And obviously this is a drawing, but yeah, you can see them in immense detail if you get them sectioned and you view them with a transmission electron microscope. So that's prokaryotes. We're very nicely going to now slip into just like almost like a bit of a tick box and add a bit more detail, just like to summarize all the different types of cells, and you'll know most of these. Go to black anyway. Animal cells, cell wall. No, we don't have cell walls. Cells, yes, we do. It's made out of cellulose. We've just done bacteria. Yes, they do. It's made out of murein, murein. And we're going to add on fungi. A fungal cell is like an animal cell with a cell wall. So I can give it a tick. It has a cell wall, and we need to know that it's made of chitin, chitin. Be careful how you say it. Chitin. Viruses, we're going to be doing about viruses. They are, as it says here, they are acellular, which means not cellular. They are not cells, and therefore it's not surprising that they don't have a cell wall. Nucleus, next. Animal cells, tick. Plant cells, tick. Fungal cells, tick, because they are eukaryotes. If you're a eukaryote, it literally means true nucleus. Prokaryotes means before nucleus, so there's no nucleus, and viruses are not cells, they definitely don't have a nucleus. Capable of self-replication. Can animal cells do mitosis? Yes, they can. Plant cells can. Again, not all of them. Brain cells can't, but we're going talking like, can any cells do it? Fungal cells self-replicate. Prokaryotic cells, they can. They don't do mitosis because they don't have chromosomes, but they can copy themselves. They do binary fission. And viruses, they cannot self-replicate. They invade cells, take over cells, and then the cell makes viruses called virions. But again, we're yet to do viruses. It's just nice to get it's like a prelude to it all. Cytoplasm, all cells have cytoplasm. Animal cells, plant cells, fungal cells, bacteria. Viruses are not cells, they don't have cytoplasm. Animal cells, yes, they have mitochondria. Red blood cells don't. Cells have mitochondria. Xylem cells don't because they're dead. Fungal cells have mitochondria. So yeah, all the eukaryotic cells are renowned for having mitochondria, and the more you have, the more energy you can transfer. Prokaryotic cells, no, they are the same size and shape as a mitochondrion. You can't put something inside you that's the same size of you. It's like putting like the same Russian doll inside the same doll. It would have to be smaller. And viruses, don't have mitochondria. They have no organelles whatsoever. Next is like, well, why am I putting carries out respiration? Well, mitochondria do aerobic respiration. So animal cells can do respiration. So it must be tick, tick, tick for those. But, can a bacterial cell do respiration? Yes, it can. If you ever did like Mrs. Gren, in like probably like year 7 or something, where one of the R's, it's like the 7 life processes, and one of the R's stands for respiration. So yeah, all living cells transfer energy. Viruses are acellular. They don't transfer energy. They therefore don't do any respiration. They just float around, and because they can replicate by getting into cells, they do. But yeah, it doesn't take any energy to do that process. The actual assembly of the virus takes energy, but that's happening in a cell using the energy that's released from respiration from inside a cell. I have to remember the order, now, because it's animals cells don't have chloroplasts because they don't do photosynthesis. There are some like weird ones like corals where there's there's like a jellyfish basically stuck onto a surface, and inside the jellyfish there'll be an algal cell, so a cell a bit like a plant cell that has a chloroplast, but again that's two things living together. Plant cells have chloroplasts. Fungi don't. They don't do photosynthesis. Bacteria, again, they are smaller than a chloroplast, and viruses are even smaller. So it's only plants, and that's like a unique one there. It's quite a nice one because of these eukaryotic cells, the plant is the only one that has it. It's its unique one. May photosynthesize. Cells don't photosynthesize. Plant cells obviously do because they have chloroplasts. Fungal cells don't. They decay things. Remember, your decomposers from the carbon cycle. However, some bacteria do. They're called the cyanobacteria. So yeah, you don't have to have a chloroplast to photosynthesize. You can kind of do it just within the cell itself. It's less efficient. But yeah, things like there's a cyanobacteria like algal bloom in one of the lakes in Northern Ireland, where it's like there's like a toxic blue-green algae. The blue-green algae are bacteria. Genetic material next. All of these do have, that they use DNA as their main one. So they use DNA is their like long term, because they do have RNA as well. Long term. I'm trying to think how to put it. There's a long term. Yeah, I guess it's just genetic molecule after genetic molecule. Viruses can be DNA or RNA. So you do get just RNA viruses, which we'll be learning about. Next, DNA is associated with proteins. Yes, it is. In the eukaryotes, it is. It's basically DNA and it wraps around histones. We do about that in topic 4. Whereas bacteria, as we just done, they don't have their DNA associated with proteins. It's just all loose all the time. All the genes are available. And viruses definitely don't. One day we're going to put a tick somewhere on the virus, aren't we? Hopefully. Anyway, we're nearing the end. Structure of the genetic material next. So in a eukaryote, they are called linear chromosomes. Chromosome is DNA. that's double-stranded, as in double helix, and it involves histone proteins. So a chromosome is histone proteins and DNA, and because it's a strip, as it's got a start and an end, we call it linear. Where bacteria, or the prokaryotes, they have a loop of DNA, and it's still double-stranded. So yeah, they have a double-stranded loop. And topic 4, we do it, but it's like there's a loop, and it will be like a double helix. It's hard to kind of draw, but hopefully you get the idea. And there's a variety in viruses. So it can be double or single-stranded. Double or single-stranded. It depends. It's good. Next, plasmids. No. Now, they have found some plasmids in some fungal cells, but it's a proper rarity. As far as our syllabus goes, plasmids are found in bacteria. Yeah. So again, it's one to kind of like have that's just for bacteria, apart from these like really weird exceptions. But maybe they're not really there, or maybe like they're due to a bacterial infection or something. So yeah, just have it as bacteria only. Ribosomes, I've got a big spot for this because they have ATS. Oops, let's stick in a black pen for consistency. Yeah, so they have ATS, which are larger ribosomes, but they do have 70S in chloroplasts and mitochondria. So it's awkward to say that they don't have 70S. Yeah, whereas prokaryotes, they only have 70S. There's no ribosomes in a virus. They just take over another cell's ribosomes. Okay, so yeah, so it is awkward, is ribosome. And then the last couple has membrane-bound organelles. We've named them. We have mitochondria. Plant cells have got chloroplasts, but there's also SER, Golgi, RER, and all that kind of stuff. Same for fungal cells. It's just like an animal cell with a cell wall. Has membrane-bound organelles? No. For bacteria, they don't have any membrane-bound organelles. The only membrane they have is their cell membrane, and there's no organelles in a virus. Then motile, powered movement. Some animal cells can. Some plant cells can. It's like moss spores, I think, or motile ones. Fungi. I can't think of any motile fungal spore. I'm hoping I'm right there. Some bacteria do, and viruses are non-motile. They don't have powered movement because they can't do respiration, so they can't power anything. They haven't got any way of transferring energy to do any powered movement. So that's that filled in, and just look at that sea of crosses for the virus. They just don't do anything. Yeah, they have nothing. Hence, when we learn about viruses, we'll see. It'll be interesting to see like what do they have and what can they do. So hopefully that was, I know the most exciting, but it's only been 25 minutes. So because it's a video, you can nip back and watch it. Sorry about my umming and ahhing, but it's better doing it like this than asking you to kind of like read some stuff and fill it in, because then it all needs corrected. At least we know it's correct and we've got all the same information. So in summary, in your DSPs and things, learn the structure of your bacteria, Again, capsule is one to learn, and the flagellum is new. And the fact that they have 70S ribosomes and plasmids, and their DNA is different. It's not a linear strand, it's a loop. Viruses, you can just leave for now because we'll be doing about them. Fungi, that's all we ever do about fungi. It's just knowing that they have a different kind of cell wall. And that's a really good, useful kind of difference in that, that they all have cell walls, the plant, fungi, and bacteria, but it's made of something different. And therefore, that's quite a good one, and one that they like to ask about because it's detailed, and you'll learn about cellulose, or you've learned about cellulose. I'm getting into waffle mode, which I do on these videos, so I shall stop now. Okay, take care. I'll see you on Friday. I'm back on Friday.